Hello, good day. So our topic for today is about the nature of antigens and major histocompatibility complex. So the reference that we'll be using is a book, Clinical Immunology and Serology by Stevens. I believe we gave you a PDF copy of this one so you can browse through it. It's chapter 3 of Stevens. Okay, aside from that, of course, I browse some informations in the internet, particularly Google. I also included the link below, especially for the figures. Okay, now prior to starting this topic, I guess it would be better that we will be discussing terms and terminologies that are common to this particular topic. Okay, so let's proceed to slide number 2. Okay, so let's proceed with key terms, or terminologies common to this particular topic. Now, before we proceed to that one, you might be wondering why I turned off my camera. Well, is it because I don't want that you will be distracted by my appearance? So I guess it would be better to turn it off. Now, let's continue or proceed with immunogens. So by definition, immunogens are any substance that is capable of inducing an immune response. Aside from that, they are macromolecules capable of triggering an adaptive immune response by inducing the formation of antibodies or sensitized T cells in an immunocompetent host. Immunogens can then specifically react with such antibodies or sensitized T cells. Okay? On the other hand, antigens refers to a substance that reacts with antibody or sensitized T cells but may not be able to evoke an immune response. So, the term antigen again refers to a substance that reacts with antibody or sensitized T cells but may not be able to evoke an immune response in the first place. Thus, all immunogens are antigen. However, all antigens cannot be considered as immunogens. But you might be wondering, well, in some books, they could be used together. Yeah, same thing with Stevens. They can use together. So, uh, in other words, we can use them, okay, at the same time. On the other hand, we also have MHC or major histocompatibility complex. MHC are the genes that control expression of a large group of proteins originally identifies on leukocytes but not known to be found on all nucleated cells in the body. So one of the most exciting areas of research nowadays focuses on how and why we respond to particular immunogens. This response is actually caused by a combination of factors. Well, we have the nature of immunogen itself, and second, the genetic coding of MHC molecules that must be combined with an immunogen before T cells are able to respond, and immunogen processing and presentation. So that's what we will be talking about. Okay, now proceeding to the next slide, another term, we have an epitope. So an epitope, this is actually the key portion of the immunogen against which the immune response is directed. This is also known as the determinant site. I will be showing you pictures of epitopes as we go along with our discussions. Okay? We also have the word haptens, which is defined as a simple chemical group that can bind to an antibody once formed, but that cannot stimulate antibody formation unless tied to a larger carrier molecule. So it means that haptens are non-pathogenic or non-immunogenic, okay, substance, but then they can become immunogenic once they are attached to a carrier molecule, like for example protein. So yeah, we'll be showing you pictures of that as we go along with our discussions. Now, here we have factors influencing the immune response. So we have five. One, we have the age, overall health, the dose, route of inoculation, and the genetic capacity. So we'll try to discuss each of them. Let's start with the age. So in general, the older individuals are more likely to have a decreased response to antigenic stimulation. On the other hand, or at the end of the age scale, like the opposite of it, for neonates, they do not fully respond to immunogens because their immune system are not completely developed. So it means that the overall health plays a role as individuals who are, number one, we have malnourished, fatigued, or stressed are less likely to mount a successful immune response. That's why it's very important that you have to take a good rest. Like for example, you need to sleep at least 6 to 8 hours a day, drink lots of water. We have 6 to 8 glasses of water. That's very important. And of course, you need to eat a healthy diet. Uh, there appears to be a threshold dose for each individual's immunogens. So this allows the innate immune response to take care of small amounts of pathogens and leave the adaptive response for pathogens that are present in large numbers. Generally, the larger the dose of an immunogen one is exposed to, the greater the immune response is. However, a very large dose can result in T and B cell tolerance. So we have a phenomenon that is not well understood. It is possible that memory cells become overwhelmed and therefore non-responsive. The actual amount of immunogen needed to generate an immune response differs with the route of inoculation as well. So like such routes includes intravenous or into the vein. We have intradermal or like into the skin or superficially into the skin, subcutaneous or beneath the skin, and oral administration, which is of course very common. That's what we've known. We have the intravenous and the very common, which is the oral administration. So where the immunogen enters the body determines which cell population will be involved in the response and how much is needed to trigger a response. Lastly, of course, the genetic capacity. So the genetic predisposition of or deposition, may be involved that allows individuals to respond to particular immunogens. Then this predisposition is linked to MHC or major histocompatibility complex, and of course to the receptor generated during T and B lymphocyte development. So these are the factors that influence the immune responses. Okay, and let's proceed. Then we have the traits of immunogen. So the ability of immunogen to stimulate a host response is dependent on these characteristics. So first we have the macromolecular size. Okay, so it means that what is the size of your immunogen. Second, we have the chemical composition and molecular complexity of an immunogen, the foreignness, and lastly, we also have the ability to be processed and presented with major histocompatibility complex um, molecules. Now, we'll try to discuss each and every one or each of these characteristics as we go along with our discussion. So first we have the macromolecular size. So it says that um, the molecular weight of most immunogens to be recognized by the immune system is usually 10,000. So that's recognizable already. However, the best molecular weight of an immunogen is 100,000 Daltons. However, I want you to remember that there are exemptions to this particular rule. So because a few substances with a molecular weight of less than 1000 have been known to induce an immune response. But however, for the most part, the rule of thumb is that the greater the molecular weight, the more potent the molecule is as an immunogen. Okay, so that is with regards to molecular size. Okay, now let's proceed to the second characteristic, which is the chemical composition and molecular complexity. So immunogenicity is also determined by substances' chemical composition and molecular complexity. Like for example, the best immunogens, we have proteins and polysaccharides. Okay, proteins are powerful immunogen because they are made up of variety of units, which is known as your amino acids. Amino acids are known to be the building blocks of protein. So the particular sequential arrangement of amino acids, we have the primary, the secondary, and of course the tertiary structure. So the primary structure determines the secondary structure, which is the relative orientation of amino acid within the chain. I believe it has been discussed in your lower years, particularly maybe in biochemistry and in cytogenetics. So just a quick review of this one. So the tertiary structure embodies the spatial or three-dimensional orientation of the entire molecule, and the quaternary structure is based on the association of two or more chains into a single polymeric chains. Now, because of the variation in subunits, proteins may have an enormous variety of three-dimensional shapes. However, these cells recognize structures that project from the external surface of macromolecules, and the more complexity or branching there is, the easier it is for B cells to respond. Aside from that, proteins have epitopes that also stimulate T cells, which is essential to generating T cell help in antibody production. Or in contrast, however, synthetic polymers such as like nylons or Teflon are made up of a few simple repeating units with no bending or folding within the molecule. So because of this reason, they are used in making artificial heart valves or elbow replacement, or could also be used for other medical appliances. So because they are considered non-immunogenic. Okay, on the other hand, we have carbohydrates, which are of course considered less, although considered less immunogenic than protein, because of the unit of sugars, which are more limited than the number of amino acids present in a protein. So as immunogen, carbohydrates most often occur in the form of glycoproteins or glycolipids. Okay, so however, many of the blood group antigens are comprised of such carbohydrate complex. Like for example, we have the type A, type B, or the H blood group antigens, which is a glycolipids, and we also have the RH, which is considered as glycoprotein. So there are other blood groups to be named, however, that will be discussed in another subject like immunohematology. Okay, so that's the chemical complexity and molecular chemical composition, I mean, and molecular complexity. Now let's proceed to another characteristic, which is foreignness. So if when we say foreignness, this is the immune system is able to distinguish between self and non-self, and those substances recognized as non-self are considered immunogenic. So this ability is acquired by lymphocytes mature in the primary lymphoid organ. So any lymphocyte capable of reacting with self-antigen is normally eliminated. typically, the more distant taxonomically the source of the immunogen is from the host, the better it is as a stimulus. So, for example, we have plant protein, which is a better immunogen for an animal than is material from a related animal. Okay. However, occasionally, autoantibodies or antibodies to self-antigen also exist. So that is with regards to foreignness. And the last characteristic, we have antigen processing. So if a molecules cannot be degraded and presented with MHC molecules, then it is considered, or it would be, a poor immunogens. The particular MHC molecule produced also determines responsiveness to individual antigens. So, for a substance to elicit immune response, it must be subject to antigen processing, which involves enzymatic digestion to create small peptides or pieces that can be complex to MHC molecules uh, to present to present to responsive lymphocytes. Okay, so that is with regards to your antigen processing. Okay, then we have the epitope. So we have here the picture of an epitope. We already discussed this maybe in the first few slides. But again, I want to reiterate epitopes. This is the key portion of the immunogen recognized by the immune response. So you have there the picture of an epitope. The green colored circle is an antigen, and the blue colored smaller circles are considered to be an epitopes. Or another representation, we have an antigen, and then the red color small ones is an epitope. And the epitope, it is the portion, a very tiny portion of an immunogen or an antigen recognized by your immune response. So although the immunogen must have a molecular weight of at least 10,000, only a small part of the immunogen is actually recognized in the immune response. So that is the epitopes. So epitopes are molecular shapes or configurations that are recognized by T or B cells, and there is evidence for proteins, epitopes recognized by B cells, which may consist of as few as 6 to 15 amino acids. Okay, now we have here the nature of epitopes. So let's try to talk about them. First, the key portion of the immunogen recognized by the immune response is the epitope. So this is a minute or small part of your immunogen. Second, epitopes are molecular shapes or configurations that are recognized by B or T cells, and epitopes may be repeating copies, or they may be differing specificities. They may be sequential or linear, or may be conformational. We'll try to see the difference between these later. And of course, epitopes recognized by B cells may differ from those recognized by the T cells. Okay, so these are the nature of epitopes. Now we have here points to remember. Uh, so first, if the immunogen is protein, B cells may recognize the primary, secondary, tertiary, or even the quaternary structure. Second, for polysaccharides, the branch points of branched chains may contribute most to recognition. And then lastly, the T cell recognize only an epitope as a part of a complex formed with MHC protein on the surface of the antigen presenting cell. And of course, for the next slide, this is the difference between the linear versus the conformational epitope. So we have here the structure. the linear epitopes, or otherwise known as the continuous epitopes, and then we have the conformational or the discontinuous epitopes. Okay, so the linear epitopes, this is consist of sequential amino acids on the single polypeptide chain, while the conformational epitopes results from folding of a polypeptide chain or chains, which are non-sequential amino acids which are brought into close proximity. So you have here the picture of the linear and the conformational epitopes. And then we have the haptens. Haptens is also important. They are considered non-immunogenic materials that, when combined with a carrier, create a new antigenic determinants, like what we have in the figure. So the red triangle are considered haptens, and they are non-immunogenic. However, when they are coupled or attached or combined to a carrier molecules, like for example a protein or an enzyme, then they can be considered as a complete antigen, and eventually they are considered as immunogenic. So that is haptens. Now, for more information, you can click the link below to know more about the haptens. Okay, and then the characteristic. These are the characteristics of haptens. Now, for the characteristic of haptens, as you can see in the illustration, the triangle, the red triangle, if it's hapten alone without antibody, okay, the antibody formation. So if it's hapten alone without antibody, it's nothing. It cannot be. It's not useful. It's not immunogenic. Okay. Now, if the haptens react with antibody, then we have that hapten antibody combination. Then, for example, we have the haptens and a carrier, then hapten and carrier complex, then there will be an agglutination reaction because, of course, the haptens in this time is being coupled with a carrier, let's say for example an enzyme or a protein, and so it could be considered immunogenic and could be recognized by your immune response. Then Then we have the relationship of an antigens to the host. Antigens can be placed in broad categories depending or according to the relationship to the host. Like for example, we have autoantigens. Those are antigens that belongs to the host. For example, my own antigens. So I have my own antigen, so that's autoantigen. Okay, like we have alloantigens are those from other members of the host species and is capable of eliciting an immune response. Like for example, in cases of blood transfusion, when you transfuse blood to another person, then your red blood cell carries an antigen, of course, and then that antigen could be considered this time as an alloantigen because it's already transferred to another host, which is another human being. Okay, then um, these are important to consider in tissue transplantation and in blood transplantation, the alloantigens, of course, because we have this what we call the graft versus host disease, in which if you transplant, or when you do transplantation like tissue transplant, and then the host does not recognize the one that's being transplanted as a self, then the host can reject that particular organ. Okay, so that's what we have or graft versus host disease. We also have heteroantigens. So these are antigen from other species such as other animals, plants, or microorganisms. So yeah, and then we also have the heterophile antigens, which are antigens that exist in unrelated plants or animals but are either identical or closely related in structure, so that antibody to one will cross-react with antigen of the other. So an example of this is the human blood groups type A and B antigens, which are related to bacterial polysaccharides. It is believed before that until now, that anti-A antibody, which is normally found in individuals with blood types other than A, we have like for example type A or type O, is originally formed after exposure to pneumococci or other similar bacteria. Naturally occurring anti-B antibody is formed after exposure to similar bacterial cell wall product. So that's an example of your what you call that, heterophile antigen. So normally in serological reactions, the ideal is to use a reaction that is completely specific, but the fact that cross-reactivity exists can be helpful for certain diagnostic purposes. So yun, that's it. So indeed, the first test for infectious mononucleosis or IM was actually based on the heterophile antibody reaction. So during the early state of infectious mononucleosis, a heterophile antibody is formed stimulated by an unknown antigen. So this antibody was found to react with sheep red blood cells, okay, and this formed the basis of bulk screening test for mononucleosis or IM. However, this topic will be separately discussed as we go along with serological testing. Okay? Okay, then we have another term, an adjuvant. So when we say adjuvant, this is a substance administered with an immunogen that increases the immune response. So the power of immunogens to generate an immune response can be increased through the use of an adjuvant. Okay, so it acts by producing a local inflammatory response that attracts a large number of immune system cells to the injection site. For example, we have an example of an adjuvant. We have aluminum salts, which are the only ones approved for clinical use in the United States, and these are used to complex with immunogen to increase its size and to prevent a rapid escape from the ano, from the tissue. So it must be injected into the muscles to work. Okay. So for example, the hepatitis B vaccination is the best example of using this type of adjuvant. Although we have another adjuvant, which is also a common one. We have Freud's complete adjuvant, which is consists of mineral oil, emulsifier, and killed mycobacterium of about 0.5 micrograms. Oh no, not micrograms, 0.5 milligrams per ml. So antigen is mixed with adjuvant and then injected. It is released slowly from the injection site. Like we have the Freud's adjuvant produces granulomas or large areas of scar tissues, and for this reason it is not used now in humans. So adjuvants are thought to enhance the immune response by prolonging the existence of immunogen in the area, increasing the effective size of the immunogen, and increasing the number of macrophages involved in antigen processing. So that is the function of an adjuvant. Okay, now we have major histocompatibility complex. Uh, for years, scientists searched to identify postulated immune response genes that would account for differences in how individuals respond to particular immunogens. So, evidence now indicates that the genetic capability to mount an immune response is actually linked to a group of molecules originally referred to as your HLA or human leukocyte antigens. We have the French scientist Dosset gave them this name. He gave this the HLA name. Okay, so because they were first defined by discovering an antibody response to circulating WBC or white blood cells. And these HLA or human leukocyte antigens are also known as your MHC molecules, okay? Because they determine whether transplanted tissues is histocompatible and thus accepted or recognized as foreign and rejected. So MHC molecules, these are genes controlling the expression of major histocompatibility uh, molecules. They are found on all nucleated cells in the body and plays a pivotal role in the development of both humoral and cellular immunity. I guess this humoral and cellular immunity has been discussed or well discussed by Ms. Merlin already. So the main function of these MHC molecules is to bring antigen to the cell surface for recognition by T cells. So in other words, um, it is the one that presents the antigen, okay, to the cell surface. Okay, we'll discuss them, and I guess there are some pictures uh, to help you understand the action or the function of MHC itself. Now let's just try to discuss first the clinical importance. There are three actually. So first you have the transfusion reaction, the graft rejection, and of course for autoimmune diseases. So these are the clinical importance of MHC or major histocompatibility complex. Well, for the main function, as I've mentioned a while ago, to bring antigen to the cell surface for recognition by T cells, this is because T cell activation will occur only when antigen is combined with MHC molecules. Okay, so clinically they are relevant because of these three reasons: transfusion reaction, graft rejection, and autoimmune diseases. Okay, now let's proceed now, for the genes coding for MHC molecules or HLA antigens, so they are found in the short arm of chromosome 6 and are divided into three categories. I guess particularly chromosome 6 P21.31. Okay, so we have class 1 molecules which coded for three different locations. We have termed the A, the B, and the C. Sorry for that. Class 2 genes which are situated in the D region with different loci. We have the DR, DQ, and DP, and then class 3 which is located between the class 1 and class 2 area, which code for the complement protein and cytokines such as tumor necrosis and factors. So for the class 2 molecules, there is a gene that codes for the alpha chain and one or more genes that code for the beta chain. Yeah, and as I've said, in between class 1 and class 2, we have the class 3 genes. Okay, so class 3 proteins are secreted protein that have an immune function, but they are not expressed on the cell surface. Unlike your class 1 and class 2 that are expressed in the surface of the red blood cells or on the cells. Class 1 and class 2 gene products are involved in antigen recognition and influence the repertoire of antigens to which T cells can respond. So let's proceed to the next slides for you to better understand. So this is the picture or of the location of the three classes, okay, of your MHC: class 1, class 2, and class 3 genes on chromosome number 6. So on my left side we have the class 1, which is divided, which codes for three genes, the A, B, and C, and on the opposite end we have the class 2, which codes for HLA, DR, DQ, and DP, and then in between the class 3, which is of course for cytokines. Okay, so again, class 1 and class 2 genes for products are involved in antigen recognition and have an influence the repertoire of antigens to which T cells can respond. Now, at each of these loci or location, there is a possibility of multiple alleles. Okay, so um, Now, when we say MHC system as polymorphic, so actually the MHC system is the most polymorphic system found in the humans or in humans. It is thought that this polymorphism is essential to our survival because MHC molecules play a pivotal role in triggering the immune response to diverse immunogens. So, again, at each of these loci or location, there is a possibility of multiple alleles, like in this picture on the screen. Okay, so the MHC system is again described as polymorphic because there are so many possible alleles at each location. For example, um, at least 580 different alleles is present in HLA-A. Okay, that is in class 1, MHC class 1. For HLA-B, we have at least 921 alleles, and for HLA-C, we have 312 alleles that have been identified. So the probability that any two individuals will express the same AHC molecules is very low because of these alleles, and individuals actually inherit two copies of chromosomes. Uh, One copy is inherited from the mother and another copy is inherited from the father, and thus there is a possibility of two different alleles for each gene on the chromosomes, unless, of course, the person is considered homozygous or has the same alleles at the given location. So, and if this is the case, then these genes are described as codominant, meaning that the allele that an individual inherits code for products that are expressed on cells. And since the MHC genes are closely linked, they are inherited together as package, which is we call as haplotype. Okay, thus each inherited chromosomal region consists of package of genes for example A, B, C, D, R, D, P, and DQ. Now the full genotype would consist of two of each genes at a particular locus because there are numerous alleles or variant forms at each loci. An individual's MHC type is about as unique as the fingerprint. So it's very rare that you will have the same MHC type because traditionally HLA nomenclature have been defined serologically through the use of battery of antibodies, and currently, however, advances in DNA analysis have made identification of actual gene possible. So the nomenclature have become correspondingly more complex. Like for instance, the notation HLA DR1 1301 indicates the actual gene involved in coding for HLA DR1 antigen. So HLA DR1 that is class 2 antigen with the B standing for the beta chain, which is part of the antigen, and the 1301 indicating the specific allele. So it's becoming more complicated, and this is because of the advancement of the technology that we have. So the uniqueness of the HLA antigens creates a major problem, however, in matching organ donors to recipient because these antigens are highly immunogenic. Okay. However, in cases of disputed paternity, polymorphism can be used as a helpful identification tool so for disputed paternity. But however, in transplantation, that's becoming a problem because we need to get the specific HLA or MHC classes. Okay, so that's the um, that's the characteristic, one of the characteristics of your MHC, which is a polymorphic one. Then we have here the structure of class 1 molecules. So you have an illustration presented to you. For more information, you can click the link below and try to browse it. However, each class 1 antigen is a glycoprotein dimer made up of two non-covalently linked polypeptide chain, and the alpha chain has a molecular weight of 45,000 Daltons. However, in some other references, it says 43,000 Daltons. And a lighter chain associated with it, called the beta-2 microglobulin, has a molecular weight of 12,000 Dalton and is encoded by a single gene on chromosome 15 that is not polymorphic. So the alpha chain is folded into three domains. We have the alpha 1, alpha 2, and alpha 3, and it is inserted in the cell membrane via a transmembrane segment that is hydrophobic. So as you can see in the illustration, alpha 1, alpha 2, and alpha 3 is inserted into the cell membrane. The cell membrane is represented by a green colored picture. Okay, so the three external domains consist of about 90 amino acids each. So 90 amino acids for alpha 1, another 90 for alpha 2, and another 90 for alpha alpha 3. So the transmembrane domain has about 25 hydrophobic amino acids along with a short stretch of about 5 hydrophilic amino acids and an anchor of 30 amino acids. On the other end, the beta-2 microglobulin does not penetrate the cell membrane, but it is essential for proper folding of the alpha chain. X-ray crystallographic studies indicate that alpha 1 and alpha 2 domain each form an alpha helix, and that these serves as the wall of a deep groove at the top of the molecule that functions as the peptide binding site in antigen recognition. Now, this binding site is able to hold peptides that are between 8 and 10 amino acids long. Then, most of the polymorphism, however, resides in the alpha 1 and alpha 2 region, while the alpha 3 and beta 2 regions are similar to the constant region found in your immunoglobulin molecules. So, the alpha 3 region reacts with CD8 and cytotoxic T cells. Then we have the MHC class 1 molecules. As I've said, they are expressed on all nucleated cells. They could be found highest on lymphocytes. However, they are low or undetected on liver, hepatocytes, neural cells, muscle cells, and even sperm. And because of this, it is explained or may explain why HLA matching is not done in cases of liver transplant, because they are undetected sometimes in the liver hepatocytes. Additionally, the HLA C antigen are expressed at a lower level than HLA-A and HLA-B antigens. So your HLA-A and HLA-B are the most important for or too much for transplantation. Then we also have the non-classical class 1 antigens. So they are designated as E, F, and G. This group of molecules, except for the G, is not expressed on cell surface and do not function in antigen recognition, but may play other roles in the immune response. While the G antigen are expressed on trophoblast cells during the first trimester of pregnancy and thought to help ensure tolerance for the fetus by protecting placental tissue from the action of natural killer cells. Now, for class 2 MHC molecules, the occurrence of class 2 MHC molecules is much more restricted compared to that of the class 1 because they are found primarily on antigen-presenting cells, which of course includes the B lymphocytes, monocytes, and dendritic cells. Aside from that, we also have macrophages. Okay, and the major class 2 molecules we have DP, DQ, and DR consist of two non-covalently bound polypeptide chains that are both encoded by genes in the MHC complex. Now, the DR is expressed at the highest level as it accounts for about one half of the class 2 molecules on a particular cell. The DRB or beta gene, on the other hand, is the most highly polymorphic as it has 18 different alleles which are known at the same time. Then let's proceed to the structure of class 2 molecules. So these are consist of two non-covalently bound polypeptide chains that are both encoded by genes in the MHC complex. Now, as you can see, both the alpha chain with a molecular weight of 33,000 Daltons, to which in some other sources it says it has 34,000, and the beta chain with a molecular weight of 27,000. However, in some other sources it says it has 29,000 Daltons, are both anchored to the cell membrane in compared to your class 1, which is it's only the alpha which is anchored to the cell membrane. But for class 2, both the alpha and the beta are anchored to the cell membrane. Now, each two domain, as it is the alpha 1 and the beta 1 domain that comes together to form the peptide binding site similar to the one found in class 1 molecules. However, both ends of the peptide binding cleft are open, and this allows for capture of longer peptide than is the case for class 1 molecules. So as you can see on the illustration, okay, so at least three other class 2 genes have been described. We have your DM, DN, and the DO, and the so-called non-classical class 2 genes. Products of these genes play a regulatory role in the antigen processing as well. So as you can see in the illustration, both the alpha and the beta domain are attached to or anchored to the cell membrane. Then, now as I've mentioned a while ago, we have the non-classical class 2 genes. These are your DM, DN, and DO. These are the product of this gene plays also a regulatory role in antigen processing. Now for these slides, we'll be discussing the main role of class 1 and class 2 MHC molecules. So their function, or the main role of class 1 and class 2 molecules, is to bind peptide within cells and transport them to the plasma membrane, where T cell can recognize them in the phenomenon known as antigen presentation. So you have there a figure to which you can see MHC class 1 and MHC class 2 molecules. So the T cells can only see and respond to antigen when they are combined with MHC molecules, while on one individual can express only a small number of MHC molecules. Each molecule can present a number of different antigenic peptides to T cells. Now, it is thought that the two main classes of these molecules have evolved to deal with two types of infectious agent: those that attack cells from the outside, such as bacteria, and those that attack from the inside. We have the viruses and other intracellular pathogens. So for both class 1 and class 2 molecules, I want you to understand that they are sensitized in the endoplasmic reticulum, and for a time they remain anchored in the endoplasmic reticulum membrane. However, class 1 molecules actually binds peptide while still in the endoplasmic reticulum. In fact, binding helps to stabilize the association of the alpha chain of class 1 with that of the beta 2 microglobulin. However, before binding with antigen occurs, newly sensitized alpha chain freely binds to a molecule called the calnexin. Now, a calnexin is a molecule to which newly sensitized alpha chain binds to. Now, this is an 88 kilodalton molecule which is membrane bound in the endoplasmic reticulum, and it keeps the alpha chain in partial folded state while it awaits binding to beta 2 microglobulin. However, when beta 2 microglobulin binds, calnexin is being released and three other chaperone molecules called the calreticulin, the tapacin, and the ERP57 are associated with the complex and helps to stabilize it for peptide binding. Peptides that associates with the class 1 molecules are approximately 8 to 10 amino acids in length and are derived from partial digestion of proteins sensitized in the cytoplasm. Now, these intracellular peptides may include viral, tumor, or even bacterial antigens. And such peptide may be newly made proteins that fail to fold correctly and hence are defective. So these peptides that are being destroyed eventually are eventually defective. So they are defective ribosomal products, or we call it the DRIPs. So 20 to 70% of all proteins sensitized in a cell may fall into this category. Now, digestion of these defective or early proteins is carried out by proteases, which will be discussed in the next slide. Okay, so this slide represents the mechanism of action on how MHC class 1 presents the antigen to the cytotoxic T cell. So as I've said a while ago, peptides that associate with the class 1 molecules are approximately 8 to 10 amino acids in length and are derived from partial digestion of protein synthesized in the cytoplasm. So we have here a picture. For example, this is the protein synthesized in the cytoplasm. Okay, so these intracellular peptides, this one, okay, may include viral, tumor, or even bacterial antigens. Now, these peptides may be newly made proteins that fail to fold correctly and hence they are called as defective. Now, these are called defective ribosomal products, or we call it the DRIPs, defective ribosomal products, or your DRIPs. Okay, yan. Let's erase this one first. Okay, now 20 to 70% of all proteins synthesized in the cell may fall into this category. The digestion of this defective or early protein is carried out by protease. Okay, so if this is now synthesized in the cytoplasm, then it is carried by proteases that resides in a large cylindrical cytoplasmic complex called the proteasome. So this is now, if this is the newly synthesized proteins, then it will be carried by proteases, okay, into a proteasome, or we call it the large cylindrical cytoplasmic complex. Now, proteasomes are actually pockets of enzyme that play a major role in antigen presentation. Why? Because peptides must be unfolded before entering the cylindrical chamber of proteasomes, and then they are cleaved into proper size for delivery to class 1 molecules. So from newly synthesized proteins carried by proteases into the proteasomes, then these should be properly folded first, and then eventually they will, or they are being cleaved into proper size for delivery to class 1 molecules. Now, for example, this is now the properly folded size. Okay, this one, the peptides. Now, once cleaved, the peptides must then be pumped into the lumen of the endoplasmic reticulum. So, this peptide should be pumped into the lumen of the endoplasmic reticulum, and that is the work of your top 1 and top 2. These are specialized transport proteins, or we call it the transporters associated with antigen processing. That's why we call it the top 1, or TAP1 and TAP2. Now, these two are responsible for the adenosine triphosphate dependent transport from the cytoplasm to the lumen of the endoplasmic reticulum of peptides suitable for binding to class 1 molecules. Now, top 1 and top 2 are most efficient at transporting peptides that have an amino acid of about 12. Okay, so 12 amino acids, they are most efficient in transporting those peptides with those amino acids or less. Now, topazine, as I've said, we have the topazine, the ERP57, and calreticulin. Now, these three are the chaperones to which when the beta 2, okay, microglobulin binds to the MHC class 1. This will happen when the MHC class 1 will release now the calnexin. Calnexin is being released from binding to MHC class 1 once your we call that 3 chaperones. Okay, once the beta 2 microglobulin is combined or attached to MHC class 1, and then when beta 2 microglobulin is attached to, like for example this one, MHC class 1, it will uh, eventually release 3 other chaperone, which is now your topazine, your ERP57, and calreticulin. Okay, now the topazine will bring the top transporters into close proximity to the newly formed MHC molecules and mediates interaction with them so that peptides can be loaded onto the class 1 molecule. So, like if this one, this is the topazine, eventually will, sorry, this topazine will eventually carry the top 1 or the transporter to carry this synthesized peptide in close proximity with your MHC class 1. So this is now the MHC class 1. Okay, now so that the peptides will eventually be loaded into your MHC class 1. Now once the alpha chain has bound the peptide, the MHC one peptide complex is rapidly transported to the cell surface. So for example, now this is now the synthesized the peptide that is now fully loaded into your MHC class 1. Now once the peptide is already loaded in your MHC class 1, then eventually the MHC class 1 peptide complex is rapidly transported to the surface of your cell membrane, of course, passing through the Golgi apparatus, then the secretory vesicle, and then eventually it will look like this. So this is now the product. This is your MHC class 1. If you noticed, only the alpha chain is being anchored to the cell membrane, and then it already presents the peptides into the cytotoxic T cell. Now remember that these defective peptides could be what again? It could be viral, it could be tumor, or even bacterial. Okay, so that is the mechanism of action of MHC class 1 on how they present the antigens to the cytotoxic T cell. Now, out of the thousands of peptides they may be processed in this manner, only a small fraction of them, maybe 1% or less, actually induce a T cell response. Now, binding is based in interaction of only 2 or 3 amino acid residues with a class 1 binding groove. Now, different class 1 molecules will have slightly different binding affinities, and it is this small difference that determines to which a particular antigens one individual will respond. Now, it is estimated that a single cell may express about 10 to the fifth copies of each class 1 molecules, so many different peptides can be captured and expressed in this manner. Okay, so then let's proceed to the next. Then we have your MHC class 2 molecules. So, unlike class 1 molecules, the MHC class 2 molecules must be transported from the endoplasmic reticulum to an endosomal compartment before they can bind peptide. I guess it will be better discussed later with an illustration that will be on the next slide. So dendritic cells, okay, these are the most potent activators of these cells, and they are excellent at capturing and digesting exogenous antigens such as your bacteria. Aside from that, class 2 molecules in the endoplasmic reticulum associates with a protein called your invariant chains. Now, invariant chains is a protein to which class 2 molecules associate in the endoplasmic reticulum. Okay, it prevents interaction of the binding site with any endogenous peptides in the endoplasmic reticulum. Aside from that, it is important to know that invariant chain is a 31 kilodalton protein that is made in excess so that enough is available to bind with all class 2 molecules shortly after they are synthesized. Okay? Okay, so this is the figure or an illustration that will better explain to us the action of your MHC class 2. Okay, so let's start with the invariant chains, as I've said a while ago. So in this illustration, this is actually the invariant chains, this one. Okay, so invariant chains may be responsible for helping to bring alpha and beta chains together in the endoplasmic reticulum lumen and then moving them out through the Golgi complex to the endocytic vesicle where digested antigen is found. So this is now your MHC class 2. Let's change the color. Let's say green. So this is your MHC class 2. So this is the beta chain and the alpha chain. And if you notice, both are present in the endoplasmic reticulum. Now that is the purpose of your invariant chains. Again, it is responsible for helping to bring alpha and beta chains together in the endoplasmic reticulum lumen, and then moving them out through Golgi complex to the endoplasmic vesicle. So this is your Golgi apparatus. This one. Okay. Now that is your Golgi apparatus. Now, because the open structure of class 2 molecules would permit binding of segments of intact protein within the endoplasmic reticulum, your invariant chain, now this one, now the invariant chain serves to protect the binding site. So if you notice, there's a small dot here now because your MHC class 2 are open. So one of the purpose of your invariant chain is to protect this site. Okay, so let's try to erase it first. Okay, so that's the purpose of your invariant chains. Okay, now once bound to the invariant chains, I mean we're talking about the MHC class 2 here. Once they're already bound to the invariant chains, okay, the class 2 molecules is transported to an endosomal compartment where it encounters peptide derived from endocytosed exogenous protein. So, like for example, this one. So, this is now endogenous proteins. Okay, phagosome. What is the cell that is most potent activator? We have your dendritic cells. Okay, so we have endogenous proteins there. Okay, now antigen processing then may help to unfold molecules and uncover functional sites that are buried deep within the native protein structure. The invariant chain is degraded by protease. So this is your invariant chain. This one is degraded by protease, leaving just a small fragment called the class 2 invariant chain peptide, or the clip. So this is now the clip. Okay, now that is your class 2 invariant chain peptide, which is attached to the peptide binding cleft. So this one. So it's very tiny. Okay, let's erase it. It's, I guess, a very light green colored one. Now, selective binding of peptide is favored by the low pH of the endosomal compartment. So if this is your endosomal compartment, if this is the endosomal compartment, it is favored by a very low pH. Okay, HLA-DM here, HLA-DM molecule helps to mediate the reaction by removing the clip. So if this is the clip, the action of the HLA-DM is to remove these clip fragments. Now, generally, peptides of approximately 13 to 18 amino acids, okay, residues can bind because the groove is open on both ends, unlike your class 1 molecules, which has a closed end. Now, within a central core of 13 amino acids, 7 to 10 residues provide the major contact point. Now, hydrogen bonding takes place along the length of the captured peptide, in contrast to class 1 molecules, which only bound to the amino acid and carboxyl terminal ends. There are actually also several pockets in the class 2 proteins that easily accommodate amino acid chains. Okay, these gives class 2 proteins, more flexibility in the type of peptides that can be bound. Now, once binding has occurred, the class 2 protein peptide complex. Okay, this one now we have the class complex is stabilized and is transported to the cell surface. So this is now the complex. So for example, this orange product, these are, for example, endogenous proteins, phagocytosed. Then the purpose of the clip. First, we have the clip from invariant proteins, class 2 molecules, the invariant proteins. The purpose is to cover the open site. Now, the purpose of your HLA-DM is to remove the clip so that it will be replaced by the antigens coming from these endogenous proteins. Now, once binding has occurred, like this one, there's already binding that occurred, the class 2 protein peptide complex is stabilized and transported to the cell surface. So, it is being transported to the cell surface and it is now being presented to the T lymphocytes. Okay, so again, this is for CD4. Okay, on the cell surface, the class 2 molecules are responsible for forming a trimolecular complex that occurs between antigen, class 2 molecules, and an appropriate T cell receptor. So these are the three, what you call that? A three or trimolecular complex. So we have here, first, this one is the antigen. Okay, sorry. This one is the antigen, and then we have the class 2 molecules, and then we have the appropriate T cell receptor. If binding occurs with a T cell receptor on a CD8 positive T cell, the T helper cell recruits and trigger a B cell response resulting in antibody formation. So in this slide, this is a simplified pictures on the action of your class 1 and class 2 molecules. So let's try to recap everything that we've learned. Okay, so for example, if this is the class 1 molecules, okay, now we have the infected cells. The infected cells, what are those again? These are your defective peptides. Okay, so the defective peptides will be digested, okay, into the cytoplasm, and then once digestion occurs, the protease will eventually carry them into a large cylindrical barrel, which is your proteasomes. Then this defective peptide should be properly folded in order to fit into your proteasomes, and then of course once proteasomes will release the peptide, what will happen? Of course, your top 1 and top 2 will pump them into the lumen of the endoplasmic reticulum. Okay, so maybe somewhere here in the endoplasmic reticulum. And then with the help of, what is that? We have your tapacin. Yeah, with the help of a tapacin, eventually, top 1 will tapacin will bring top in close proximity with your MHC. Of course, the top is the one that will pump the peptides or digested peptides into the lumen of the endoplasmic reticulum, and then tapacin will bring them in close proximity with your class 1 molecules until such time that, okay, for example, this is your class 1 molecules until such time that this antigen fragment, what is the one that carries them? It's your tapacin that carries them with the aid of TAP, of course, in close proximity with your MHC class 1, and once it's loaded, then your class 1 will be transported into the surface of the cell, and then eventually your MHC class 1 molecule will present it to your cytotoxic T cell receptor. So we have here, if you notice, okay, for your MHC class 1, it has something to do, it's associated with CD8. Okay, these are MHC class 1 are usually virally infected or mutated cells, so it could be a tumor cells. Why? Because these are what again? Defective proteins, right? So then they will present that to the CD8 or cytotoxic T cells. And then of course, degradation will happen. Now with your class 2 MHC on the other hand, what is the most potent T cell activator? We have your dendritic cells. Okay, for example, the dendritic cells will capture the microbes. So it could be, these are exogenous proteins. We have, it could be bacteria. Okay, so once they phagocytose it, eventually these endogenous proteins will be digested and eventually will be transported into endosomal compartment, and it will stay there. Now, for example, this is, okay, so this is now your exogenous proteins carried into endosomal compartment. Then we have here your MHC class 2 with the help of what again? Invariant proteins. Okay, the invariant proteins will carry alpha 1 and alpha 2 into throughout the Golgi apparatus into endosomal compartment, where they will encounter the exogenous, okay, proteins, and eventually what will happen? It's the clip, or we have the class 2 invariant chain peptide attached to the peptide binding cleft here. Okay, now the clip, the invariant chain, okay, with the help of the clip, will cover the open system of your, let's erase first, we'll open the open system, will cover the open system, because this is the open system of your MHC class 2. Now, once it's already covered, so we already have a cover here, with the aid of what is that? HLA-DM, it will eventually remove the segment of invariant protein so that the antigens or the exogenous peptide will be loaded into the MHC class 2, like this one, like the picture here. Now once it's already loaded, eventually the MHC class 2 will be transported into the surface of your dendritic cells per se, and then the class 2 molecules will present this exogenous molecules into the T cell receptor, which is usually your CD8 or the T helper cell, like this one. So the professional antigen presenting cell this time is, for example, the most potent, which is the dendritic cell. So CD8 or T helper cell. So the thing that you need to understand is that if it is class 1 molecules, it will eventually present defective proteins, those that will produce tumors or virally infective cells or mutated cells. But for MHC class 2, these are those antigen presenting cells that phagocytosed bacteria, for example, or other microbes. Now, these are points to remember with regards to MHC. So, as few as 10 to 100 identical antigen MHC1 complex can induce cytotoxic response. Second, in healthy cells, most of these MHC class 1 complexes contain self-peptides that are ignored by the T cells, while in disease cells, peptides are derived from viral proteins or protein associated with cancerous states. Now, display of hundreds of class 1 molecules complexed to antigen allows CD8 positive T cells to continuously check cell surfaces for the presence of non-self antigen. Now, if recognized an antigen as being foreign, then the CD8 positive T cell produces cytokines that cause lysis of the entire cell. Okay, so for the next 3 slides, I've get this from the internet. These are the major differences between the MHC class 1 molecules and MHC class 2 molecules. This is with regards to characteristics. So we have the distribution, the encoding genes, the nature of antigen presented, and the antigen itself. So you can browse through it and read all of this. Aside from that, other characteristics would also include enzymes involved in peptide generation, peptide loading of MHC. So I guess this has been discussed a while ago. Peptide loading complex. Okay, so we have for MHC class 1, we have the top 1 and top 2 with the help of topazine, ERP57 on the cult reticulin, which are the chaperone molecules, while for MHC class 2, we have the chaperone and endoplasmic reticulum is the invariant chains. Okay, recognizing co-receptor for class 1, we have the CD8, and then for class 2, we have the CD4, and then for receptor cells, okay, CD8 T cell receptor for 1 and 2 for CD4 T cell receptor. Then we have the structures here, so you can just read through all of that. And lastly, we have the building blocks of our building amino acids. For class 1, it's 8 to 10. Class 2, it's 13 to 18. Peptide binding domains. So we have alpha 1 and alpha 2 are peptide binding domains. Because why only alpha 1 and alpha 2 are anchored to the cell membrane? While your MHC class 2, it's both alpha and beta are peptides are the binding domain because they are the ones that's being anchored with the cell membrane. Invariant chain is only present in MHC class 2. Okay, functional unit, we have presence of abundant antigens, target cell for destruction, that is for 1, and for 2, presence of foreign antigens, induced antibody production. And then lastly, the detection method, we have serologic testing. Okay, thank you so much. Then let's proceed to the next slide. I believe this is the last one. which is the clinical significance of MHC or major histocompatibility complex. So both class 1 and class 2 molecules can induce a response that leads to graft rejection. Second, MHC antigen plays a role in development of autoimmune diseases. And then we have the class 1 and class 2 molecules plays a major role in antigen presentation. Aside from that, the presence of particular MHC proteins may confer additional protection. Okay, so thank you so much. I believe this is the end of the discussion. And so again, thank you so much for listening. I hope that you've learned something from this discussion. Again, the reference that I'm using is Stephen's Clinical Immunology and Serology by Kristen Dorstein Stevens. So you can browse to it. We are on chapter 3. Aside from that, of course, credits to the figures that I get from the internet. So bye bye and see you next time.